The amount of information we're storing these days is going up and up exponentially. But the methods we're using for storing information are getting less and less permanent. A hard disk drive that's used in a major data center around the world has a working life of about 3 years. So when you upload your information to the cloud, or you Facebook, or when the NSA in America takes your information against your will and starts analyzing it, behind the scenes there's a manager who is spending his or her whole life working out a system for how do you replace all those disk drives every 3 years without losing any information. At best, it's a massive management of information, and at worst, things get lost. So I was talking with some colleagues one day. We were at a meeting about our research institute and how we store our data. We have a responsibility at the European Bioinformatics Institute to store data for the whole world, to add value to that by cross-referencing things, and to serve that back as a community resource for research scientists around the world. And we were in a bar in Hamburg after a day of meeting, worrying about how we would be able to afford to keep on storing this exponentially increasing amount of information that we're asked to deal with. And we wondered, isn't there some other way we could do it? And of course, the other thing I work on is evolution. And in evolution, we're very much aware of a system which stores digital information and passes it on for millions or hundreds of millions or thousands of millions of years. And it's the DNA in the genome of every living organism. So we wondered, is it actually possible to use DNA, the chemical molecule DNA, in itself? It's a chemical molecule which can hold information. It consists of 4 molecules, 4 different types of molecules. They're shown here. Typically, all the scientists in the world know them by the letters A, C, G, and T. So we have some chemistry on the right-hand side, and in the middle, the famous double helix structure that DNA forms. That keeps it really, really stable. It lasts a very long time, as long as it's not too wet or too hot. And the double helix is this famous thing that means it's why it works so well for transmitting information, because the process of making copies is very easy. You split the 2 parts of the helix apart, and then each one acts as the template where you can make another copy to make 2 copies, and then 4 copies, and then 8, and whatever your body, the cell, needs those for. The information is actually not in the helix as such, but either single strand of the helix has all the information in it. Once you have one part of the helix, you kind of know what's going to be in the other one. It's very, very small. So 4 angstroms, 10 to the minus 10, 4 times 10 to the minus 10 meters. It's tiny, tiny, tiny. To have a system to store and recover information, what do you need? You need to be able to read it, you need to be able to write it, and you need this language. We also can't make very long fragments of DNA. We can't make a genome's worth of DNA. We can't make enough DNA to hold a video or a PDF or some piece of digital information. But we can make short pieces, and we can make a lot of short pieces, so we can split our message up. Reading DNA, well, we're much better at that. That's been going for many years now, and of course, everyone, I think, is familiar with genome research programs. The human genome was first sequenced in the year 2000 at a cost of approximately $3 billion US. Nowadays, you can do a genome. That took years, a concerted effort across the world. Nowadays, one lab can read something like a human genome in a day or 2 at a cost of maybe $1,000 or $2,000. Can we do an experiment where we take digital information, any digital information, anything you like, anything on my laptop computer, we use some code to convert it into something that looks like DNA. We get the company to actually make that DNA. They ship that to us because they're in California, so we know they're going to have to send it to us in a box. They use FedEx. It comes back to us, we take it to the laboratory, and we say to the laboratory guys, can you read this back? Does this work? Has this worked? And we see if we can recover the message. The thing that's missing is the code. We need a code. How do you take digital information and turn it into something that you can represent by a molecule of DNA? So what we did have to put a bit of work in ourselves was create a code, but we had to work out a code so we would start with binary information. On the computer, it's represented as 0s and 1s. So we had to invent an error-correcting code, just like your mobile phone or digital TV or your hard disk in a computer. They're all doing error correction. They don't tell you about it because no one really wants to know there's errors going on and being corrected. They're there, they're happening all the time. If you've got the right code, you can deal with it. We invented a code that would deal with the kind of errors we expected in our DNA experiment. So we had our plan, we had our files, we have our code, we have a technology to write and read DNA, and that all worked. And they FedExed a box back to me, and I opened it on my desk, and there's these tiny test tubes with almost nothing in. I thought they were actually empty, and I had to show my colleague, because I was trained as a mathematician, so I don't know what DNA really looks like. It looks like nothing. I took them to my colleague, and I said, Paul, they haven't sent any DNA. And he said, no, it's in there. Look, and you hold it up to the light, which is what that picture is, me holding up to and you can just see this kind of speck of nothing in there. And that's enough DNA to store about 1 megabyte of computer files. So we now have sonnets and photographs and PDFs in a test tube of actual DNA. It's not in a living organism. We're not using living DNA out of the genome of a living thing. There's ethics issues with doing that, and it's very difficult to keep a cell alive if you're manipulating it in this way. We're just using the chemical DNA. And we took it to our laboratory colleague. We used our decoding procedure, which is what the graphic at the bottom is representing. The answer is, yes, it did work. And it worked 100%. Not just that the pictures looked the same and the PDFs looked the same. Every single byte of information, every bit of every byte of about 1 megabyte of information was perfectly reproduced. And a lot of other companies are interested. We've kind of invented this field of research now, and these other companies are wading in that are interested in doing this because there are a lot of advantages if we can make a working, viable, everyday system out of DNA. So it's coming along. We're very optimistic that in 3 or 5 years, these will be viable technologies that anyone could own. Why would you use DNA for storing information? It's tiny. The amount of information you get in a certain volume is huge. So 1 megabyte fits in something you can barely see. In real terms, if I'd filled that test tube, so it's the size of 2 joints of your little finger, that much DNA would be equivalent to 1 million CD-ROMs. What it means in real life is you don't need things as big as data centers. You can get everything in the whole world in the back of a medium-sized truck or an SUV. You can't afford to do that yet, but that's the kind of space it would take up. Here's another advantage. DNA lasts a very long time in easy-to-create conditions. This will work for a 500,000-year timescale. And that's quite a bold statement. I avoid making bold statements, but I'm allowed to make one bold statement like that because I spent the preceding 20 years studying evolution. And we have sequenced DNA sequences from mammoths, from Neanderthals 40,000 years old, from bison at 60,000 years old, and from ancient horses over 500,000 years old. The DNA from those living creatures has survived in a readable form for that time scale. And those weren't carefully prepared scientific samples. Those were just dead things that happened to die somewhere that was very cold. If you want to make copies of DNA after the first copy, so I told you the first copy is hard. Once you've got the first copy, the second and the subsequent ones is really easy. The machine on the right-hand side it's called a Pcr machine. Every laboratory has one of those. It essentially goes through a cycle of heating and cooling the chemicals in a certain way. It's copying the process that goes on in living cells of the way DNA is replicated. And it just takes a few minutes to make 2 copies. And then it takes another few minutes to make 4, and then 8, and then 16. And once you've got 1 copy of the information, it kind of self-assembles more and more copies in an exponential way. And you can easily make millions of copies of your original information in just minutes or hours. As long as there are people with technological interests, they will be people who want to read DNA, and we will have a machine to read DNA. The machine changes. It's gone from being a laboratory to a thing on the desk to a thing you can hold in your hand. In another 3 years, it will change again, but there will always be a DNA reading machine because we all have DNA inside us. It's part of our makeup. It's part of what explains how we age and how we get ill. And as long as we want to improve our own health, we will be reading DNA.